Hello learners and welcome to chapter 9 where we get to focus on muscle and muscle tissue. So our goal is to figure out what is muscle, what does it do, and how does it do it. So what we get to learn is we get to learn about muscle and muscle tissue, the structure of skeletal muscle from a gross anatomy perspective, which is what we see here, to a microscopic perspective. Then we're going to talk about, well, how do muscles contract? That's going to be our understanding of the sliding filament theory. And finally, we'll end with developmental aspects of skeletal muscles. Where are skeletal muscles generated from an embryonic standpoint? So let's focus on the properties of muscle tissue. Muscle tissue is specialized for contraction or to create movement. At the cellular level, there are different things that can actually cause movement, but at the tissue level, that's the focus here, the tissue level, muscle tissue is the only thing that can move, okay? And muscle is going to move in one way. So muscles move in one way. And they shorten. Going to shorten or contract with force. So that's how muscles move. And for this to happen, we must convert chemical energy to mechanical energy. So chemical energy is going to be the energy stored in ATP, right? And then that ATP energy is going to be converted to mechanical energy or movement. So every time you see mechanical energy, just think movement as well. Cool about this, folks. Every time you're dealing with muscles and the movement of muscles, you're going to be generating heat. And that's how we maintain our body temperature when it comes to homeostasis. So, of course, the properties of muscle tissue are very important to help us be able to take ATP energy and create it into movement and generate that heat to maintain body temperature. Now, remember from our previous chapters, we have types of muscle tissue, right? But all those muscle tissue is going to share the same properties. Okay, so here are the properties we have: contractility, extensibility, elasticity, and excitability. Let's focus on contractility. That means you're going to forcibly shorten a muscle. It means that the muscle is going to get shorter with force. So here we have our bicep, and then we also have our tricep here. When we actually bend our arm, then we are shortening the bicep. And when we have our arm relaxed, we are going to be shortening the tricep. Next, we have extensibility. And just think extend. That's going to be the ability for the muscles to stretch. So we already talked about contractility. That means that you are going to shorten the muscle with force. But if you pull on a muscle, then it's able to get longer. And that's where the extensibility is going to come in. So you see this person that's doing a yoga pose here. A lot of stretching and contraction, right? So we want to make sure that the muscle can return to its original size, giving rise to the property of elasticity. So you see this rubber band here, you can stretch it and it'll go back to its original size. When you stretch or contract a muscle, it returns to its original size as well. Another property that's very important. We have to be able to think about how that signal to contract is going to be given to muscle. Well, here we see we have a brain and we have a muscle. That means that we have a nervous system and muscle connection, right? And that is going to be excitability. And that means that you're able to transmit stimuli. So we see we can have a situation where the nervous system is transmitting stimuli to the muscle, but guess what? The muscle itself is able to transmit stimuli as well. That means muscle cells can also pass stimuli with those action potentials. And the membrane of muscle cells are excitable, much like the nervous system. So it's important for the muscle cells to know when they're supposed to contract. And that's how we're able to see that excitability process. So in order for contractility to happen, you have to have the ability for the muscle cell to have excitability. They kind of go hand in hand. And you ask yourself, how do muscle cells get the signal to contract? How do they spread the signal? How do they shorten and contract with force? That's going to be based on contractility and, of course, the excitability, being able to transmit that stimulus, which we get to talk a little bit about in the rest of the lecture. First, let's review the 3 types of muscle tissue in the human body. Of course, our focus is going to be skeletal muscle, but we'll talk about cardiac and smooth muscle in this table. To ask, how are these 3 tissues similar or different? And how we do that is going to be based on location, where they're located in the human body, whether or not they're voluntary or involuntary, are they striated, how many nuclei per cell, and also we're going to see it visually through a microscope, and that's going to be our light microscope. That's going to help us make some distinctions. All right, the 3 types: skeletal muscle, cardiac muscle, smooth muscle. Our focus is going to be skeletal muscle. Now, the location in the body. Skeletal muscle is connected to bones. Cardiac muscle is heart. Location is in the heart. And smooth muscle is going to be our blood vessels and hollow organs. Some examples of these hollow organs are going to be our digestive tract and the bladder. Okay? Now let's get into voluntary, involuntary. What are we really asking here? Can you consciously think about contracting these muscles? Well, with skeletal muscle, yes, you can voluntarily say, hey, I'm going to move my bicep, and I'm doing it now, right? That's me voluntarily thinking about it. But when it comes to your cardiac muscle and your smooth muscle, they're involuntary. So you don't want to have to think about it, right? You don't want to be dealing with your smooth muscle and say you are eating food and it's going down your actual esophagus and you're telling yourself, okay, keep moving down, keep moving down, keep moving down until it reaches the stomach. You don't want to have to do that. So that is going to be involuntary. Cardiac muscle is also going to be involuntary because you definitely do not want to have to tell your heart when to beat, how many times to beat, et cetera, et cetera. So when it comes to smooth muscle, which is made up of hollow organs and blood vessels, sometimes folks have a hard time understanding the involuntary portion. Well, just think about it. With the hollow organs, like our digestive tract, you're like, okay, just like we talked about, the food is going down the esophagus. I'm not telling it when to go down or push, push, push. It's doing it on its own because of the overall squeezing and pushing of that particular smooth muscle. The blood vessels, again, I'm not telling my blood to move around in my body. Again, that's going to be the squeezing and pushing of that smooth muscle of the blood vessel. Now, when we talk about the bladder, sometimes it can cause confusion for folks. So the bladder is definitely a smooth muscle. That means definitely it's going to squeeze and push when necessary. And so what happens is when it's time to pee, your bladder is going to squeeze. And it has inner valves in it that's going to open automatically. You don't have to think about it, okay? But in order for us to actually have the urine go out, we're going to need help of skeletal muscles. And the skeletal muscle is going to be like a sphincter that's at the bottom of our actual bladder, and also our abdominal muscles can tighten and help compress the pelvic cavity where our bladder is going to be sitting to kind of finish getting all that pee out. Just think of like when you're holding pee, you're holding it not because the bladder is doing it involuntarily. You're holding it because the skeletal muscle sphincter that's at the bottom of the bladder is holding it. But the fact that you're trying to pee, that's all involuntary. Now let's get into striations when it comes to muscle types. Now a striation is basically just a stripe-like pattern, or you can also say a crosswise pattern that shows up on the light microscope. So we'll see and talk about light microscopes here. Now when it comes to skeletal muscle, yes, it is striated, and you will see that pattern below. Cardiac muscle is also striated, although most of the time when you're thinking of cardiac muscle, you're going to be looking at branching, and then it'll be harder to find the striations within that branching. We'll try our best to see some here. And then finally, we have smooth muscle, which is going to be non-striated. That's going to be the easiest one to distinguish because it looks so different. There's no striations, there's no branching. Again, it's going to be made up spindle-shaped cells that we know are going to be long and skinny and have that fatter middle. And it has no crosswise pattern. All right, let's go ahead and look at the light microscope. So here in the light microscope, of course, it is going to be hard to see. Skeletal muscle is the most pattern that we see with the striations. You see striations here. I mean, you can just see the cross sections here, striations. All through, all through, all through. You see it going all through here as well. More striations going along. And this is going to be a microscopic view. And yes, we'll talk about more details of what these striations are and really be able to understand that these are part of sarcomeres, which are then portions of the microscopic making of a muscle cell. Okay? So again, what do we see? A bunch of striations. That's going to make it very easy to understand. Then when it comes to cardiac muscle, it's striated, but mostly we're looking at branching. So we start here, we see branching, branches out here, here. Also we have branching here and here, right? Another thing you might notice is like these little lines. So let me move that. You might notice these like little lines here. So here's a line here, here's another one here, here's a couple more here, here's some down here. And some folks will be like, oh, well, that obviously the striation. No, these are going to be called intercalated discs. Intercalated discs, I'm just put ICD there. And these are just linking a series of cardiac cells to one another. The striations are going to be the hardest to see on the cardiac muscle cell. Of course, you can go ahead and look at your text and have enlarged photos Of course, you can also go ahead and Google it to see different types of images, or just go back to our original where I gave you that link of all the different microscopic images when we talk about histology and look at cardiac muscle there. Like you can see some striations here, can see some striations up here, but the most thing you're going to notice is that branch-like pattern when it comes to cardiac muscle. And you have to just tell yourself, yes, it is striated in certain areas. Now, smooth muscle, again, spindle-like cells. And this is going to, you're going to see that these are just going to be in a smooth muscle type pattern. There is no crosswise pattern. There's no striations, nothing like that. Just look smooth, smooth, smooth as you're going across. There's no branching, anything like that. Okay? The last thing we can focus on when we're trying to distinguish is going to be our nuclei per cell. So nuclei per cell means how many nuclei are within the cells of these muscles. So the muscle cells are all going to be the same length. That means however long your bicep is, the muscle cell is going to be that length. And so since it's very long and you have to fuse a lot of these cells together, they're going to need a lot of nuclei. So you'll see the nuclei here in purple, nuclei here, 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 here. That's a lot of nuclei because, again, what are we doing? We are generating heat with movement. So we're taking ATP energy and we're going to convert it to mechanical energy. Guess what? That's all going to take a lot of nuclei. And again, we need to make sure we can get oxygen, so it has to have blood vessels and everything going through it. And we also need nerves going through it if we're getting signals from the brain. So yes, they're going to need a lot of nuclei per cell in order to fulfill their overall function. Next, we have cardiac muscle. And yes, you do see probably mainly one nuclei per cardiac cell, but usually it's going to be 1 to 2. Some of them are binucleated. That's where that 2 comes from. And so I definitely can see a nucleus here, maybe 1. I can also see a nucleus here, here, and you also see some nucleus that are beside it. Sometimes that's endothelium as well, so just keep that in mind when we're looking at cardiac muscle. And then finally, we're talking about smooth muscle. They're also going to have 1 nuclei. So you can see that this is 1 nuclei here, and that means that we're talking about 1 spindle cell. You see a bunch of them grouped together. That means they're each individually a spindle cell. Another spindle cell here, 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 and here. So they're all going to be individual as well. All right, folks, so this is going to be our introduction to muscle and muscle tissue. Next, we're going to talk about the structure of skeletal muscle, both gross anatomy and a little bit of the microscopic anatomy. And then by the end of the entire lecture, it's going to be all focused on microscopic anatomy. See you there.